The movement of water is a special type of diffusion that is referred to as osmosis. And in the body, we are very concerned with where water will move, because water is one of those few substances that we really cannot control the direction that it moves. It follows a general trend, and that is to move towards wherever there are more solutes or things dissolved. Now, the way that we measure the concentration of solutes is in millimolars or micromolars, and then collectively we call them osmoles. This is discussed more in my workbook in chapter 11 and also in lab 1. So I'm not really going to revisit that here. But we're looking at the total concentration of solutes or stuff. My two compartments are going to be my extracellular fluid and my intracellular fluid. My extracellular fluid has a distribution of blue solutes. My intracellular fluid has a distribution of reddish solutes. Now notice that the concentration of those solutes is approximately equal. In other words, the concentration of solutes here is about equal to the concentration of solutes there. They have the same density. So therefore, they have the same osmolarity. Now, I'm always interested in which compartment is going to have a greater osmolarity, or maybe they have the same osmolarity. And so the way that I describe that is with this sentence. I'm going to let my extracellular fluid be my starting point, and I'm interested in the concentration of stuff in my extracellular fluid relative to the concentration of stuff in my intracellular fluid. Is my extracellular fluid have a greater concentration of stuff? Again, it does not matter what the stuff is. Does it have the same concentration of stuff, or does it have a lower concentration of stuff? And so we denote those comparisons by using these prefaces: hyper, iso, and hypo. So if the extracellular fluid had a greater concentration of stuff, I would use this prefix, and I would say the extracellular fluid is hyperosmotic. Hyper means more than, excessive, or greater. It turns out that the extracellular fluid actually has the same concentration of stuff, the same osmolarity, so we use iso to say the same. So the prefix that's going to make this sentence correct is iso. The extracellular fluid is isoosmotic to the cell. Now, something important about osmolarity and comparing two compartments and determining which of these prefaces you're going to put in front of the word osmotic is that we are simply looking at a snapshot in time. We are looking at a picture. We are saying right now, where's the concentration? Is it greater in the extracellular fluid? Is it greater in the intracellular fluid? Or is it the same? That alone does not tell us which way water is going to move. We are simply saying which compartment has more stuff at this very moment. Let's take a look at another example. Which one of these prefaces makes this sentence correct based on this picture? Well, if I take a box, I notice that my extracellular fluid has a greater density or concentration of stuff, both blue and red, and my intracellular fluid has a lower concentration of stuff. So clearly my extracellular fluid has more than my intracellular fluid. There's less. So to complete this sentence, I am going to say that the extracellular fluid has more stuff than the cell, and therefore it is hyperosmotic. Let's look at another example. Which prefix are you going to put in the sentence to make this statement true? Hypo. The extracellular fluid has less stuff. Now, the solutes are concentrated inside the cell. So here's where we get into water movement. Water likes to move to where there's more solutes. So based on this image, you might predict that water is going to move from the extracellular fluid to the intracellular fluid. And when it moves to the intracellular fluid, it's actually going to make that cell expand out as it gains water. Much like a water balloon, as you fill it up, continues to expand outwards. And the problem with our cells, our human cells, is that our plasma membrane is kind of flimsy like a water balloon. And if it fills up with too much water, it can actually lyse. So you can end up with a lysed cell, where the cell basically explodes, much like a water balloon. So we need to be a little bit careful and make sure that we don't let too much water in. So water likes to go where there's more solutes, but the osmolarity does not tell you the entire picture. Based on what I have shown here, you would predict that water is going to move into the cell. In this image up here, you're going to predict that there's going to be no net movement of water. Water will move, of course, but we won't see any appreciable changes in cell volume. And then in this one, we actually expect that water will move out of the cell, and the cell, as a consequence, is going to shrivel up. Okay, that is all true if the solutes cannot cross the plasma membrane. So this is a really important consideration. But if the solutes can cross the plasma membrane, then the osmolarity will not alone tell you which direction water will move. So while osmolarity gives you a snapshot in time about the differences in overall solute concentration, they are only a piece of the puzzle if the solutes can in fact cross the plasma membrane. So let's take a look at what I mean. Consider this scenario. I have here a cell that contains a certain concentration of potassium, and my extracellular fluid contains a certain concentration of sodium. And I have drawn my intracellular fluid and extracellular fluid so that the concentration of potassium equals the concentration of sodium. So as I've drawn this, the extracellular fluid is isoosmotic to the cell or the intracellular fluid because they have the same concentration of stuff. And so based on this drawing, you might say, well, there's no net movement of water because the concentrations of everything are equal. But what if I tell you that I put in a bunch of potassium channels? These are going to be channel proteins specifically for potassium. Now we have a solute, potassium, that is allowed to move across that membrane, where sodium is not. So, because we are looking at the snapshot in time as shown, I would still say that the ECF is isoosmotic to the cell. But I actually predict that water is going to move out. Interesting, huh? So why is that? Since I've inserted a bunch of potassium channels, I expect that potassium is going to move until it reaches its own diffusion equilibrium. That is to say, the concentration of potassium inside the cell will be equal to the concentration of potassium outside of the cell. Because I do not have sodium channels drawn here, sodium will stay concentrated in the outside of the cell. So I expect something like this. Well, now you can see that once potassium is allowed to exit through those channels, water is going to follow the solutes as they move. So water will also exit, and therefore water will move out of the cell to the extracellular environment. The way that we describe the predicted movement of water is through something called tonicity. We use the same prefixes that we used for osmolarity. We still use iso, hyper, and hypo. So if there is no net movement of water, I would use iso. If the movement of water is from the cell to the extracellular fluid, I'm going to use hyper. And if the movement of water is from the extracellular fluid into the cell, I'll use hypo. So here, water is going to follow potassium out of the cell, and so this is hypertonic. It's going to result in my cell volume shrinking. So here you can see that osmolarity and tonicity are not always the same thing. Osmolarity, again, is just the initial snapshot, how much stuff is in one compartment versus another compartment, and does not take into consideration that some of the solutes may move. In order to predict tonicity, we have to know which solutes can move. And tonicity is really the descriptive term that tells us which way water is going to move. So let's take a look at another example. In this example here, I have my intracellular fluid. I'm going to let my purple dots be potassium. I'm going to let my red dots be sodium. And then the blue hexagons are glucose. So let's count everything up. Inside of the cell, I have 1, 2, 3, 4, 5, 6, 7, 8 potassiums, and 1, 2, 3, 4, 5, 6 glucose molecules, for a total of 14 things. We'll just call it stuff, but really it's solutes. Over in my extracellular fluid out here, I likewise have 1, 2, 3, 4, 5, 6 glucose molecules, and 1, 2, 3, 4, 5, 6, 7, 8 sodiums. So if I ask you to fill in this sentence, what are you going to say? And your correct answer is iso, because there's 14 stuffs in approximately the same volume of space, so that's the same concentration of stuff, and 14 stuff in here. But now I'm going to ask you to predict which way water will move, if any. And in order to predict that, we need to know which way solutes are going to move, if any. So you'll notice that by itself, neither potassium, sodium, or glucose can diffuse across the phospholipid bilayer. They are all going to need some help in transport. They're going to require some sort of protein-mediated transport if they move at all. Well, it turns out I do have a transporter, a carrier protein. And this one is in fact a co-transporter. This is the sodium glucose co-transporter. So the way this works is sodium is allowed to diffuse as long as it brings its glucose with it. So how many sodiums are going to want to come in the cell? And in order to answer that, we need to look at sodium all by itself. So I'm actually going to highlight sodium for you. There are 8 sodium ions. So if sodium is allowed to reach diffusion equilibrium, I expect that 4 sodium ions will want to come into the cell. But the only way that they're allowed to come in is if they bring a glucose molecule with it. So let's go ahead and see that in action. So here's 1. Here's 2. Here's 3. And here's 4. And I'm just going to move these out real quick to spread them around. So which way did solutes move? Solutes moved into the cell, and therefore water moves towards the direction of more solutes. Since they started out with equal concentrations of solutes, we've now moved some solutes into the cell, and therefore water is going to want to follow. So in this case, as I've drawn it, water will move into the cell will swell, and therefore the extracellular fluid is actually hypotonic to the cell. One more thing before we end. Notice that only 4 sodium ions moved into the cell. How come this one didn't move in, or this one? Remember that the way the sodium glucose co-transporter works is that sodium is allowed to reach diffusion equilibrium. And as I've drawn it, sodium has. We have 4 sodiums on each side. It's now at diffusion equilibrium. So sodium is no longer compelled to come into the cell, and therefore it does not. And without sodium driving the transport, there's no glucose following along as the unwilling friend. All right, that concludes this lecture and our lecture series looking at molecular transport in human physiology.